Well, hello everyone, and thanks for joining our April 2026 ABV webinar at DataGene. It's a pleasure to be with Pete Thurn today to provide some updates that relate to the upcoming ABV release. And there's two main topics that we've got today. One is about the stillbirth breeding values that are new to our evaluation. And the second is an update to Jersey type that Pete will explain a little bit later. We're going to kick off with the stillbirth component and then move topics halfway through. Please feel free to drop a note with some questions, or if you feel comfortable and want to interrupt and ask questions as we go, I don't mind that at all. So it's great to hear your questions, and if I haven't explained something very well, it's great to stop and just make sure that we cover things so that we're all across it. So Pete, if you're okay, we might kick off. Lovely. Yeah. So this might be a funny picture to start off a presentation about stillbirth in cattle, but bear with me for a minute. For the farmers around the group, I'm going to dare suggest that if that tractor broke down every second Sunday morning, you probably wouldn't call it just bad luck. You'd ring someone and get the tractor fixed. And similarly, if the milk truck driver turned up and reliably left about 5% of the milk in the bottom of the vat, I'm pretty sure there'd be a quick phone call made to the milk supply company to make sure that that is fixed up. We wouldn't just describe it as something that happens or bad luck or that's just farming. So my question is, why is it that we are willing to accept losses, and what I think are too many losses, as just part of farming or just bad luck? I'd like to share some findings with you, and these are findings from two different research projects at Dairy Bio. One was led by Majid Cancerfed that looked at the survival of cows in the lactating herd, and the other was a calf vitality project that I had the good fortune to be able to lead over the last few years. And this, if we look at our herd as a herd of 100 lifetimes, the thing that happens is we lose about 5% of those calves at birth or within the first 24 hours. They disappear someplace between the calving pad and the calf shed. We lose another couple before weaning. We lose some more before those animals' first calvings. I don't know if we have a good number for that, but we'll just call it 2% for the sake of the argument for today. Before the second calving, we lose another 20 animals. And before the third calving, there's another 16 to go. So by the time we get to the third lactation, we've lost a little over half of the animals that we started with. Now, I think that all of those animals represent an opportunity. And we can sort of accept these losses as kind of bad luck or natural attrition, or that's just farming, or we can actually hunt for some productivity improvement that comes from lower mortality in our cow and calf herd. In terms of the size of the prize, when we think of calves in particular, you know, that lost calf, it could have been maybe a $1,600 export calf. Maybe it's a $2,000 heifer at a dairy sale. It's also, those lost calves represent, you know, poor welfare outcomes, lost genetic potential, and I think wasted effort. It took a fair bit to get that calf on the ground to start with. It's unfortunate that it died. So, can we do a better job of using the genetic tools that we've got at our disposal to breed animals that thrive in our dairy systems? The brilliant thing around calving is that while we've got this reliable pattern of cows calving routinely, and this is the distribution of calvings by date in Australia over the past kind of decade or so, you know, not only do we have a special thing happen in terms of a cow ready to lactate and a calf ready to become the next replacement in our herd, but we also have a record, a calving record and a calf record. And these records are super important when it comes to thinking about genetic evaluation. So when we record that the cow calved, and we record the calving ease and the calf size and the sex of the calf and the date, that initiates a calving record that we can use for genetic evaluation. And one of the things that comes with that is whether the calf was born alive or dead. So we've got a data system that's set up to be able to help us to improve stillbirth. In addition to that, I had the good fortune to work with about 50 farmers around the country in the Calf Vitality Project, where we collected the genotypes of both alive and dead calves, about 20,000 calves in total, plus health events, things that happened to them during their young lifetimes. And this information was helpful for us to be able to develop new breeding tools that are the Stillbirth ABVs. One of the things that we learned about the fate of calves when we looked at this group of 20,000 calves with good health records is that the most frequent reason as to why a calf dies is because it died at birth or shortly thereafter. This is the charges of frequency of deaths in our calf vitality data set, and that big brown bar you see on day 0 are our stillborn calves. And that really outweighs the deaths that were due to scours or respiratory disease that we often think of as being really problematic in the calf shed. But in big terms, while they're annoying, they occur at lower frequency compared to stillbirth. In this data set of 20,000 calves, we had the chance to look at some incidents of various diseases, which I think people sometimes find interesting. And we've got this for both Holstein and Jersey animals. I'll just start with that first column. And if we look at the Holstein calves, we see that about 6% of those calves had a case of scours, and about 1% of them died from that case of scours. Less than 1% had respiratory disease, and by the time we get to about 12 weeks of age, we've lost about 2% of the calves that were born alive. And the stillbirth rate is just a little low, below 5%. Similarly, for Jerseys, we see similar numbers, maybe a little bit higher pre-weaning mortality, a few less scours, a little bit higher stillbirth, but very similar numbers really. The averages do hide some of the story though. So if we look at the next column, these are the maximum incidence of these diseases in herd year cohorts. So in some herd year cohorts, we had in Jerseys, 18% of them having scours, 8% of them dying before weaning, or 16% born dead. And the same is true with Holsteins. Those upper limits are a little bit scary. And what that means to me is that, it's worthwhile hunting for some improvements to be able to reduce the rate of calf health issues, including stillbirth. So people sometimes ask what influences stillbirth? Why are calves born dead? And some of the things that we found to be important is one, the age of the dam. So those animals that are having their first calf, they are at higher risk than the animals, the dams that have calved for in the second or more calving. Calving ease is a significant contributor to stillbirth. Those calves that are born with difficulty are obviously at much, much higher risk of dying. There are differences, as I showed in that table before, between herd years and seasons. And so some seasons are worse than others, some months are worse than others, some years are worse than others, and some herds are worse than others. And so, there are differences between those herd year season groups. The Jersey calves are a little bit higher risk, so there are differences between breeds. The sex of the calf is important. So male calves are at higher risk than females. Twins are also at higher risk than single births. Gestation length is also useful to consider. Either too long or too short increases the risk of stillbirth. If we've got some hypocalcemia, so milk fever in the dams, that increases the risk of stillbirth. And there are many other factors involved. It's a fairly complex biological thing, this whole calving process. And so there are things that can go wrong. So we use our understanding of the things that influence calf death to help us build a prediction model that can be effectively used then to rank animals for their ability to either deliver more or less dead calves. And in the case, the model that we are introducing here in Australia is a multi-trait model that uses information both from the stillbirth trait and also calving ease. And that helps us boost the power of the prediction model. We adjust for some fixed effects, and the fixed effects in this case are herd year season, the parity of the dam, so how many calvings the dams had, and the sex of the calf. And the evaluation is done within breed. Now, for the parents out there, I suppose one thing I learned pretty early on with my kids is that I had way less control than what I thought I would have. And the same is true when it comes to calving. It is a team effort to have a calving occur with both the calf and the cow involved in that calving process. So it makes sense to me that we have two stillbirth breeding values. One is focused on the calves that are born alive. So this is our sire stillbirth ABV. So it predicts the percentage of progeny that are born dead. The average, the way it's going to be expressed in Australia, the average is 0 and lower is better. We want less stillborn calves. So if you saw a number of -3, that suggests that there's 3% fewer calves that are born dead or die within a few hours. The maternal stillbirth ABV is a separate ABV, so you'll see two different numbers for a bull or for a genomically tested animal. That maternal stillbirth ABV, it predicts the percentage of calvings that result in a dead calf. So this is from the dam's perspective, the cow's perspective. Again, the average is 0 and lower is better. So if you saw a -3 for maternal stillbirth, that's 3% fewer calvings producing a dead calf. With the test data that we've got available, we've been able to kind of look at some genetic trends over time. So what's changing over time? And this is the genetic trend for sire stillbirth for Holsteins. And we can see that it actually is heading downwards, with the most recent sort of number being, -2%. And this is a good news story. It means that the breeding practices that we have already are making an improvement for sire stillbirth through the kind of indirect routes of other traits. So calving ease would be a prime example. The work to improve calving ease is likely having an impact, a positive impact on stillbirth. So that's a good news story. Doesn't mean that we can't improve further, though. In the case of maternal stillbirth, for Holsteins, we also have a positive story in that the maternal stillbirth breeding value, it continues to go down, and that's a good thing. These are the similar graphs for the Jersey breed. The average is very close to kind of 0 at the moment, and if we look back, back to 1990, the average, was just a little over 0, maybe almost 1, and we're down to -0.6. So we haven't sort of seen the same improvement over time with Jerseys, and that probably makes sense. There hasn't been the same focus on calving ease in Jerseys because that's something that naturally the breed has been good at over time, but it's probably good to kind of just watch this trend and see if we can still make an improvement to reduce stillborn calves in the Jersey breed. And a similar story in terms of maternal stillbirth. So soon, we'll have 2 new breeding values that you'll see in the various kind of, Good Bulls app and on DataVat and the places that you receive breeding values. In terms of what to expect, most of the bulls, I'll talk about bulls in this case, most of the bulls in Holstein will be between kind of -2 and +2 for both breeding values. Now, if we're going to look for really good bulls, the numbers that you're probably looking for are, a bull that's -4 or below. The range in Jerseys is slightly narrower. The standard deviation is about 1.5 or so instead of 2. So there's a bit narrower range, but you're still looking for bulls that are, in the -3 or -4 sort of area if we're looking for extremely good stillbirth bulls. And that's actually true for both the sire stillbirth trait and the maternal stillbirth trait. Sometimes people are interested in what this trait is related to. And we can compare the breeding values across a bunch of different traits to see where there are either positive or unfavorable relationships. Now, it's not a true genetic correlation. It's an estimated genetic correlation based on ABVs. And so if we compare ABVs that we are know and familiar with, so our indexes, for example, BPI or SI or the health weighted index HWI, we see that there's a favorable relationship between the stillbirth traits and the indexes that we're familiar with. There's also a favorable relationship, as you would expect, with calving ease. In addition, survival ABV and gestation length and somatic cell count are also favorably related. Now, just to expand a little bit, shorter gestation length is in line with lower stillbirth. We probably can't go too far there, but if we look at the breeding values, there's a positive relationship between gestation length and stillbirth. And lower somatic cell count is also favorably connected to lower stillbirth. I can't explain the relationships, but those are the way that the relationships occur. On the opposite side, if we look at things that are not favorably related, some of the size traits, so things like chest width and body depth, are negatively related with both maternal stillbirth and sire stillbirth. So the first release of these breeding values in our routine release is actually not too far away. It's expected on the 24th of March. Now we're still going through testing procedures, but at this point it's expected about the 24th of March. And that puts us in a situation then to release them publicly on the 9th of April, which is, I think, the public release, the first public release date. So a few kind of notes to think about and maybe share with your neighbors or your colleagues or the people that you're working with in your organizations. Every animal that's born is an opportunity. And I think we've now got some new genetic tools that can help us increase herd life and reduce the case of stillbirths. This has been a nice partnership between Dairy Bio, producing the research and delivering it to Datagene in a way that we can then share it with farmers that they're able to use the product of the research in making on-farm decisions. And I suppose it would be remiss of me not to remind people that, in order for us to do this work, data recording and the active genotyping females is really important for strong evaluations. And so just thinking through opportunities that you might consider in terms of how we might be able to improve that data recording is really helpful. I'd like to take a minute just to thank especially my colleagues at Agriculture Victoria through the research process and my colleagues at Datagene who were super helpful in migrating through the whole research process as well as achieving successful implementation at Datagene. And also the funders of the research whose logos are down at the bottom there. None of this research would happen if we didn't have farmers that are contributing data, farmers that were participating in the calf vitality project, herd recording centers, breeder organizations who are all helpful in making sure we have this data, and the genomic service providers for helping us to genotype animals. I'm going to stop there for some questions with just a little cartoon to have a look at, but keen to kind of take on any comments or questions that people might might have. I'm not sure if there are any that have come through, Pete, so far that you've spotted.
Not at this stage, Michelle.
Terrific.
But if people do want to ask a question, if they're adept at typing, they can pop it into the chat, or they can take themselves. Or you can unmute themselves.
Unmute yourself and say hello too. We're happy to chat that way too.
Yeah, just one question, Michelle. The drop in the sire stillbirth ABV, so it was quite noticeable. Would you put that down to the improvement in calving ease of Holstein over that period?
Absolutely. That's
okay.
my first instinct from a couple of perspectives. One, calving ease is actually part of the stillbirth model, so it makes sense that the two are strongly related. And we think about in that time period, there's certainly been quite a focus on improving calving ease. We've had better calving ease breeding values because we've got genomics included in those evaluations, so we can make better progress. So I think that's a logical explanation for that change. I'm also interested because the timing is also consistent with daughter fertility, and I'm also interested in the interaction between stillbirth and fertility. We know that, for example, animals that have had a difficult calving producing a dead calf, those dams, they are more likely to produce less milk in their first lactation, and they're more likely to have difficulty in rebreeding in that lactation as well. So I can't unpick the connection, but to me, there's something that's worth investigating in terms of that connection with fertility.
Interesting, because the recent focus on particularly haplotypes and genetic defects, that's only been in the last, a focus probably in the last 3 to 4 years. So we probably haven't seen any improvement or much improvement from that yet, have we?
I don't think so. And it's not something we're able to pick apart in this, by looking at it a breeding value, because a breeding value is sort of a collection of all those reasons why, animals could die. But there are certainly some nasties out there. One of the last papers of my research, we looked at haplotypes and we looked at particular SNPs, particular places on the DNA strand that had the potential to reduce stillbirth, and there are some ones that have quite significant impacts. The problem is there is over 200 of them, and so it's kind of hard to manage over 200 of these things. So at the moment they're sitting in the paper, but not something that's easy to incorporate into a breeding program. And that's why I think probably a good way to start is by using the stillbirth ABVs as a way of putting some downward pressure on stillbirth.
Michelle, I also thought when I saw those graphs that particularly in the Holsteins, with the massive improvement in fertility at around that 2010, 2012 era was when we had the most gain. So I sort of thought that bulls like Oman and that might have had a fairly significant impact in that trend line. But I was surprised that so many bulls sort of fit between that -4 and +4, or am I reading that kind of graph wrong? I thought there would have been a greater variation in the spread.
Yep, so let's go back and have a look at that. So this is the graph. So we started this is back in 1990. So just to explain, sorry, Pete, can I just double check that we are seeing this sire stillbirth? Yep. Okay. Yep. I changed, and I just want to make sure that it changed for everyone, not just for me. So in 1990, this is the average breeding value of bulls born in 1990 that were Holstein, and for the traded sire stillbirth. And so it was, say, about 2.5%, and we're down to -2. So to me, Daryl, I think the faster way to make genetic progress is to breed for the trait itself directly. And so while we're having some indirect improvement here, we probably can make faster gain if that, but by being able to breed directly for the trait rather than indirectly through the others. Yeah. I'm not sure if I understand the question. I'm not sure if I completely got a great answer for it. to know why we haven't made more improvement. But I think going forward, it just makes more sense to me to be able to breed for it directly rather than indirectly.
I think it's been one of the success stories of genomics is the way without seemingly a whole lot of sacrifice, we've been able to improve calving ease in Holsteins rather dramatically, to the point, now where you just don't see a lot of bad calving ease bulls in the marketplace. And then the flow-on effect of that has been what we see here through stillbirth. And I was only talking to farmers yesterday and we were lamenting the days of, Donor and Showtime and Southwind and Windluke and a whole lot of bulls that, are down in the low 90s for calving ease, which those bulls just don't get to the market these days. And the flow on of that is more live calves. And like Michelle says, I think once we start putting positive selection pressure on stillbirth as a trait on its own, we'll see much greater gains in this space.
Thanks,
and the other interesting comment with that too is that when you look at that movement over time, particularly in Holstein, our levels of inbreeding have probably increased over that same period. So, you might expect that as the level of inbreeding increases, Michelle, that you might see more issues. But so far, it's, the level of inbreeding doesn't seem to have pushed it the other way.
Yeah, I did look at inbreeding in the group of 20,000 calves, and I couldn't detect a significant difference between, in terms of calf mortality or calf morbidity between highly inbred or lowly inbred calves in my study. So I think that still quite understanding that is, there's still some work to do. Any other questions that people have?
The inbreeding component is probably more around the fact that we're concentrating more of the good genetics. Inbreeding from a negative point of view about having more stillbirths would be if we were inbreeding with more haplotypes or more detrimental genes. So the whole argument of inbreeding is there's many sides to that story. We can't just say that inbreeding is a bad thing, because essentially by concentrating all of the traits that we want to have, we are going to naturally increase inbreeding to some degree.
Yeah, the tricky bit is that especially in the recent inbreeding. So I think old breeding is maybe not so much the issue. Old inbreeding is not so much of an issue, but recent inbreeding I think is more likely to be an issue. And when I found a couple of hundred haplotypes, right now we'd name them 1, 2, 3, 4, 5, 6, 7. When you start talking about kind of a couple of hundred haplotypes for a single trait, it becomes really quite unmanageable, and that's really where I think controlling inbreeding becomes more powerful. Is that it's just impossible to kind of look at the haplotypes individually when you get to have so many. So keeping that cap on inbreeding, still seems to be a useful thing to do over time. Thanks, Jamie.
Sorry, Michelle, can I just ask, you've spoken a lot about averages and trends. In terms of outliers, are there individuals in particular that are extreme outliers, even for, negative in terms of high levels of stillbirths, and same as an extreme outlier for very low levels of stillbirths? And are there any sort of characteristics that these individuals might have?
Yeah, so I can answer part of that question. So yeah, you will find -9, -10. So there are some extreme animals out there, as you would expect. The standard deviation is about 2, but even if you move out 3 standard deviations, there's still going to be outliers at the very end. I haven't looked at those animals individually to kind of make any story about why they're so terrible for stillbirth. That's not something I've done quite yet, but you will see bulls that are extreme in terms of 9s and 10s.
So one thing, Michelle, that gave me comfort when I did the some of the QA work on some of the test runs, and there's maybe not many people, though there'd be one bloke, Pete Williams, who'd be old enough to remember this bull. There's a bull called Brystar that was a bull that was imported from the US, whose calves had arthrogryposis type issues when they were born, and he is about the worst Holstein bull for stillbirth. So when I saw that particular bull right at the bad end, and knowing the history of that bull, I thought, yeah, good, this makes sense. Everything's pointing in the right direction. So
Thanks, Pete. Thanks, John. I might pass it across to Pete now, but please feel free to shoot through any questions to any of the team let us know if there's something that we can do to help. There will be some stillbirth resources available on the DataGene website in the coming period that will have a summary of the presentation for you and for your colleagues. Thanks, Pete.
No worries. Thanks, Michelle. Okay, so I want to talk today about some changes we're making to Jersey type breeding values, and this has been something that's been in the pipeline for a little while, probably ever since the Jerseys updated their class system to include composites or new composites for feet and legs, rump, and dairy strength. When we think about type, we have come a long way. I found, when I was doing a bit of Googling, I found this wood carving from 1898 of a Jersey cow, and I thought, my God, when you compare it to the cow Jeff Akers is there, I think we have certainly come a long way when it comes to improving type of jerseys. But I think some of the changes that we're making in this next release will help that. So we've had the data come out of the New Jersey class system for some time now, and as I said, we've now got composites for dairy strength, rump, feet and legs, and mammary. Prior to this, we just had overall type and mammary. Now we have overall type plus those other 4 composites. So what's changed in Jersey type? Well, previously, the old Jersey type, overall type, was, as it come out of the Jersey classification system, was a function of general appearance, head, conformation, and vessel. It's a term you don't often hear, but I suppose back in the old Jersey world, others used to be vessels. And the old Jersey memory system was previously calculated by us using a formula, and it included the traits udder texture, fore attachment, rear attachment height and width, centre ligament, and front teat placement. So there's some missing parts of the puzzle in all of this, and we'll highlight some of those as we go through. The new composites, and this is the data that we get from the Jersey Society. You can see the breakdowns of all the composites there. And you can see just for example, in memory system, there's a lot more traits involved in memory system now. So we're including teat length, for example, and rear teat placement. I'm just having a bit of trouble reading because it's very small on my screen. In this new fancy presenter mode in Teams. So you can see we've had an additional couple of traits come in there, and then the other 3 composites have basically been put together based on the New Jersey class system. So all that data now is flowing through into the genetic evaluation system, and we're able to produce breeding values that more reflect the New Jersey class system. So New Jersey overall type, and we can compare the new versus the old there on the same thing, on the same graph, is around 40% mammary system. Sorry, 40% dairy strength. I'm really struggling to read this, it's so small. 35 mammary system, 10% feet and legs, and 15% rump. So you can contrast that with what we had previously, and you can see there's Laura's going to help me here. Maybe. That's all right. It's a good thing I can remember what I put in my screen. Resume from slide 7. Yes, resume. That's a bit better. Thank you. Bit of technical assistance with the new system. So yeah, we can compare the old and the new, and so the thing about this is it's had a flow-on impact into overall type, as well as not just adding in the 3 new composites that we haven't had before. We've actually got the new composite for overall type and the new composite for mammary. So here we can compare what we have now with what we had in the past in terms of just the mammary composite, and you can see we've added udder depth, rear teat placement, teat length, and those have been added at reasonable weight as well. So they're now starting to have an influence on the mammary composite and obviously down the track bull rankings. So what to expect? So we're going to see revised ABVs for overall type and mammary system. We're going to see the new ABVs for rump, feet and legs and dairy strength. We're also going to see some of the more extreme bulls are going to ease back for overall type and mammary ABVs. So but overall, there's also some general movements improved. So overall type correlation between old and new is 0.7, and mammary is 0.83. So, and I'll show just some scatter graphs so you can get a bit of a feel for the level of proof movement. But you know, correlations of that magnitude would indicate that there's going to be a reasonable amount of re-ranking of bulls for type. So just brace yourselves for April, because there's going to be a little bit of change in this space. One thing that doesn't change much though is reliability. So the big data set of bulls that I looked at, reliability goes down by about 1%. So there's very little change in terms of reliability. So I've de-identified this group of bulls, but there's some savvy people on the call I've noticed, and I'm sure some of them are going to know which bulls which in the scheme of things here. But this is the top group of bulls on type, and you can see the sort of changes there. So if it's a pink square with a red number, it's a negative change, and if it's a yellow square with a green number, did I get them right? Did I get those colours right?
Yellow and yellow.
A yellow and yellow, is it?
Yeah.
I'd have no idea on the green and red.
I know you're with me, Daryl. We're one of the 8%, I think.
It's just yellow.
Yeah. So if it's yellow, it's either gone up or it's neutral, and if it's pink and red, it's gone down. So you can see at the top end, more have gone down than have gone up. And like I say, some of those extreme bulls are not quite as extreme as they used to be. When we think about it, you know, and I remember when the New Jersey class system was introduced, there was a lot of commentary around 2-year-olds not scoring as high as they used to. This is pretty much a reflection of that. You know, the data that goes in, the type data that goes in, yes, we munch it up and we turn it into a breeding value, but at the end of the day, it is very reflective of the phenotypic data that we're collecting. And so if 2-year-olds aren't scoring as high, it stands to reason that type proofs won't be quite as high either. So the overall type changes, you can see here that there's quite a scatter of bulls. So it's not the same trait as it used to be, and the 0.7 correlation tells us that. And there's probably a little more change at the top end of the population than there is at the bottom end. And unfortunately, as you know, we're always looking at the top end of the population. If we look at mammary system, it's similar but not quite the same. The correlation is better, but again, there's going to be some re-ranking of bulls, and so really we'll see the first glimpse of this at the routine run prior to the public release in April, which was the 24th. So you'll get your first taste. The bull companies will get their first taste of these changes on the 24th of March, and then that will flow through to the public release in early April. So that's pretty much it from me. Happy to take some questions. Yeah, and just by the way, if any of the bull companies want to have a one-on-one session with their field staff or their team, more than happy to do that, and we can maybe even go into things in a little more depth than what we have today, and potentially share some of your results.
Are there any questions for Pete?
I'm not sure if it's a question or a comment, and I'm not sure whether this is a reflection of the way the jerseys have weighted it, and you've taken that data, or how DataGene have done it. Probably hoping for a little bit more change, particularly in the memory. I just think, like, a memory, in my mind, should be a bit about a management, or ability to manage. And if you can't put the caps on and walk away, then the bull's not worth 115 for memory. I just wonder whether we've still got enough weighting on some traits that, like, if the teeth are too short or aren't in the right place and they cause management problems, I can't see how a bull can still get to 112 or 115 for memory.
I'm not. I mean, that I'm going to tread carefully around your comment, Daryl. So one of the things that we're just starting down the path of is, even though we haven't even introduced these new composites for jerseys, but we are investigating type composites as a whole. So not just for jerseys, but across the board. And, you know, with the idea of maybe trying to attack some of the anomalies that we often see within type composites, and some of those anomalies are the kind of things that you're alluding to. The other thing that we've got to particularly understand here is, even though there might be weighting in the jersey or in a breed society's classification scores, when we convert those things to a breeding value, we have the byplay of the correlations between traits coming in. And so even though a trait might have, say, 10% weighting in the class score, when we convert those to breeding values and we start to look at the direction traits tend to go in, that breeding direction can be hampered by its negative correlation to another strong trait. So, you know, we've got to think about that, and that's part of the work that we're doing currently, looking at composites overall, is whilst there may be an intent to move a trait in a particular direction through the class system, when you get it into the genetics world, because it has a negative correlation with another trait, it erodes that potential genetic progress that you can make. So we're just at the stage of starting to understand that. And, you know, some of the teat traits probably fall into that basket, you know, where there's some funny correlations between teat length and overall memory, for example, and other memory traits that kind of counter the effect of having a positive weighting on teat length in the class system.
Well, I think it was well answered, Peter. I think the other side of it too is, in a lot of ways, they're two separate things. The weightings in the class system is around how close that animal is to ideal. But when we're looking at composites and even individual traits within breeding, as you say, you've got those correlations, but we're also looking at, from a composite point of view, is where does the population actually sit and which direction do we need them to move to become more ideal? So it's two quite different things between those weightings, or between a class system and between composites as well. So they can't really be looked at as being the same, which is difficult for, I guess, for people to get their head around, but that is the difference.
Yeah, that's right, Jamie. And the other complication we have in this space is, and some of you probably have seen the intermediate optimum expression that we've adopted, but a lot of the traits that we want to put into composites have intermediate optimums, not extreme optimums. So we have to come up with ways of managing that within the calculation of a composite, which, when we take it outside of the class system, becomes a harder task, because at the moment the Jersey classifier is the one that is managing the intermediate optimums for us. Whereas if we were to move to a more of a formulaic calculation of composites, we would have to find a mathematical way of handling that, which is not insurmountable. It just becomes another little complication.
That's what'll keep you in a job till you retire, Pete.
We got any more questions? No, if not, Michelle, have you got any final comments?
No, not for me, except we've got some key dates that are important. So the 24th of March is when we see the first routine release of the updates that we've talked about today. And then the 8th of April, did I get that right? The public release of April ABVs, that people will see it more generally. And in the meantime, if you've got some questions, you want to go through this in greater detail, or you want to share information with your colleagues, either through resources that are on the web or from a face-to-face conversation, please reach out to us. And thanks for the opportunity to share this information, the updates with you. Thanks very much, Pete, for being a great co-host.
No worries. Thank you, Michelle. And thanks everyone for joining us.